after his talk. And the other announcement is about next week's speaker. Next week, speaker will be Mala Maini from University College London, who is working on the novel immunotherapy for the HBV and HCC. So please join us again. So now I'm very happy and honored to introduce Hiroshi Ohno. For his introduction, Hiroshi is Dupli director and also team leader of the laboratory for intestinal ecosystem of RIKEN Center for Integrative Medical Science, where I also belong. And he's also professor at Chiba University and also Yokohama City University. And he graduated from the School of Medicine, Chiba University in 1983. And I'm surprised about this, but after 4 years of experience as anesthesiologist, he got the PhD from Chiba University and become a scientist. And so let me change to see the CV. Okay. And he spent 3 years as a visiting fellow at NIH USA, and he was appointed as a full professor first at Kanazawa University before joining to RIKEN in 2004. And Hiroshi is immunologist and studying intestinal immunology. Especially, he did fantastic work on the M cell, which is a unique intestinal epithelial cell subset. And he's also studying the host gut microbiota interaction to understand the impact of gut microbiota on the host physiology and pathology. He have over 200 original articles, and it is including nice paper with a high citation. And I want to say congratulations for his purple medal from our emperor last year. Which means he did he's a very famous Japanese immunologist and did many fantastic study. So now, Hiroshi, if you are ready, could you show up to our video? Okay, yes. So congratulations for your medal.
Thank you.
And thank you very much for joining to this Global IMMUNOTALK. And before moving to your talk, we used to have one question for the speaker. And my question for you today is, what is the advice you would give to your 25 to 35 years old yourself?
That's a very kind of difficult question, but so first of all, thank you very much for very nice introduction. And actually, as Kazuyo introduced, I spent 4 years in immunology and then entered the PhD course. So, and then I went to US mid-30 and came back from US almost 40. So I kind of, at that time, I kind of regret that I should have entered PhD right after graduating from med school. Then, because in several ways, you are kind of older than the same experienced scientists. But at the same time, if I looked back, maybe those 4 years of the clinical experiences could also be sometimes helpful for me. So it's not straightforward answer, but I think anything that you experienced in your past could help you later on. So that's my kind of what to say impression when I ask that kind of question. Is it okay?
Yeah, thank you very much. Yeah, but finally, you made a great success, so I think your choice was correct.
Thanks.
Yeah. So, if you're ready, could you share your slide? And
Yes.
I heard you're gonna talk something about the lung, not intestine today, about asthma. Okay.
Well, okay.
It's
Can
not
you
the
see?
slide mode. Yeah, it's not the slideshow mode.
Yeah.
Yeah, now,
Now?
okay, perfect.
Okay.
So, could you
So,
please start your presentation? Thank you.
okay. So thank you very much for this opportunity for organizers. I'm going to talk about this thing. And okay, so several slides are kind of introductory slide. So as you may know, compared to the human somatic cells that are making your body, it's reported to be like this. So our body hosts significant numbers of bacteria, both outside skin and also mucosal surfaces. I omit in this slide the respiratory organs, and especially in the colon, it is reported that 40 trillions or more bacterial cells reside in the body, in the gut, the colon. So it exceeds yourself cells. And I think many of you know that thanks to the emergence of so-called NGS, next generation sequencer, now you can know which kind of bacterial microbes are there in a given environment, including in your gut, by fully sequencing them. So there are two different methodology. One is this shotgun sequencing, where you extract and fragment the DNAs from a given environment, such as the feces, and then you can sequence deeply those, whole genome DNA. And then you could know virtually all organisms in a given environment and all the genes in those consortia of the organisms. Another one is the 16S RNA gene amplification sequencing, where a part of 16S RNA genes, these are the schematic view of this 16S RNA gene, and the thin line is a kind of universal part. So almost all the bacteria have the same or very similar sequence here. And then those boxes are the variable region from 1 to 9. So if you design the primers like here and here, or here and here, and especially degenerative primers, then you can amplify virtually all bacteria of this region, and then sequence those amplicon, then you could know which kind of bacteria are there and how many. And this is because the sequence, whole total sequence length is shorter, so cheaper. And this is longer, so more expensive. So you can choose which to do. For example, all the samples by this, and then you can pick several samples to more deeply investigate. Okay, and after these, and also the comparison of the so-called healthy people and those patients suffer from various diseases, then many scientists reveal that not only gastrointestinal diseases that are more intimately interact with the gut microbiota, but immune diseases, metabolic diseases, and neuronal diseases, the fecal microbiota composition of those patients are significantly different from the healthy controls. And I'm going to talk about allergy today, and especially asthmatic condition. And the strategy we take is not only the genome sequencing that I just told you, because the genome is just a catalog of genes. So you don't know how those genes are transcribed and translated protein and those. So in addition to the genome or metagenome analysis, we also employed epigenome. Oh, spelling it different, but so epigenome is you know that how those gene expression is modified or regulated, and then as a result, by transcriptome analysis, then you can identify which genes are made to the RNA and how many, and then proteome to know how many different proteins are made, and then metabolome, so how many small metabolic molecules, metabolites are made. So, and those, cyclopedic, different layers of the cyclopedic analysis are combined, and then multivariate association analysis are done to know what's going on in your gut and what's going on with this gut microbiota and interacted with our body. And another strategy that we are taking is the utilization of the germ-free mice. So the mouse embryos are taken out from the uterus by cesarean section. Then the uterus environment is sterile. So you can treat pups in this sterile condition and then put into this kind of the germ-free isolators. Inside this isolator is also sterile. So you can keep the mice sterile, and you can even bleed the mice in the box. And then Notabio technology is to those mice, sterile mice, you can put one or two of your interested bacteria, and then in this closed environment condition, only mouse and the bacteria of your interest is the living organisms in here. So those are not a biotechnique. So utilization of these multi-omics combined with this, we are doing, we are studying the gut microbiota and host interaction. So today's topic is done by Dr. Takashi Ito, who used to be in my lab as a postdoc, and he now went back to be a medical doctor in Chiba University. And it is published in Gut Microbes. So the strategy is this. So so-called Chiba high-risk birth cohort allergy cohort is done by mainly in Chiba University and Chiba Medical Center. So they, actually Professor Shimojo's group, recruited 306 pregnant mothers. It's a, I said it's high risk because the either mother or father has a allergic history. So that Dr. Shimojo called it high risk birth cohort. And unfortunately, 37 babies withdrew, but 269 newborns, we collected feces periodically like this. And then at age of 5, Dr. Shimojo's group diagnosed whether they have bronchial asthma or not. Of course, the bronchial asthma prevalence is not so high, but like a 10% or 50%. So we had 23 asthma group and rest is non-asthma group. Then we compared first the fecal propionate, acetate and butyrate, those short-chain fatty acids. These three are the major short-chain fatty acids and actually major metabolites among all the metabolites made by bacteria, gut bacteria. And we found that only in 1-month-old feces, propionate is tend to be low in asthma group compared to non-asthma group. And acetate and butyrate are not so different. And one of the potential source of this propionate could be the breast milk. So we compared the concentration of the propionate and also acetate and butyrate in mother's breast milk of the asthma group and non-asthma group, but in the breast milk, the concentration of propionate is not so different. So we thought that this propionate came from gut microbiota, and in the asthma group, even though the diagnosis was at 5 years old, but even way before 1 month old, the asthma group gut microbiota produce significantly less propionate. And we also compare the propionate level in the feces with the fecal microbes, and we found that several microbes, butyrate asthma group is positively correlated with propionate. But we only know that, and we don't know the significance or consequence of a causal relationship of this propionate and these groups. Excuse me. So this is the pathogenesis of bronchial asthma taken from a review article, a little bit modified. And eosinophils are known to be the major player of bronchial asthma, and also Th2 cells and Th2 cytokines. And then so we examine these in the mouse model. So we cannot modify the newborn mouse gut microbiota to produce less or more propionate. So we decided to feed their mothers right after birth in the drinking water, addition of acetate, propionate, or butyrate separately. And also another group is control water, just the water. And then 3 weeks later, they're weaned. So only the 3 weeks mother fed these waters. And then after weaning pups, the drinking water is just the water. So only in this duration, the pups could have a propionate from mother's milk. And it was the case that, as you can see here, that the fecal propionate in propionate fed group, the propionate concentration is significantly high compared to control water or acetate or butyrate fed pups of the mothers fed acetate or butyrate containing water. And then after 2 weeks, so when they are at 5 weeks or so, house dust mite are given to them to induce airway inflammation resembling bronchial asthma. Then this is the inflammation score in the lung. So you can see that propionate group, fed group, significantly low inflammation score. Acetate group also a little bit low, but propionate is very low, significantly. And then when examine the bronchoalveolar lavage cells and also lung tissue cells, then again, in the propionate group, the eosinophil infiltration in the lung and also the propionate appeared in the bowel, significantly a low number. But neutrophils and CD4 T cells are not, it's kind of vary, but not significantly less, both in bowel and lung tissue. So this indicates that the propionate in the lactation period could affect and prevent the asthmatic airway inflammation induced by HDM. And it is likely involve the eosinophil like this. And we also measured the HDM specific IgG1 and mesenteric lymph node type 2 cytokines, IL-5 and IL-13, and here again, propionate significantly suppress the serum HDM specific IgG1 and also the type 2 cytokines in the mesenteric lymph node. So these suggest that propionate in the breastfeeding period can suppress later on the HDM-induced airway inflammation. And as you may know, the GPR41 and GPR43, two G protein coupled receptors, are well-known receptor for propionate and also acetate and butyrate. So we next examine whether GPR41 or GPR43 is involved in this propionate mediated prevention of the airway inflammation. And you can see here that this is the inflammation score in the lung. So only GPR41 knockout cancel the propionate feeding effects, but GPR43 knockout suppressed by propionate of the airway inflammation like wild type. So 43 is not involved, but 41 is it. And consistently, HDM specific IgG1 also suppressed with in the GPR41 knockout like wild type, but in the GPR41 knockout, it is also cancelled. And these are also the similar results that bowel eosinophils and lung eosinophils. Again, the GPR41 knockout, the suppression is cancelled, and neutrophils and CD4 T cells are not involved in this as expected. So GPR41 is important in this experimental setting. Then, because actually this is asked by reviewers that mother, GPR41 absence itself could do something, but not propionate. So we cross the GPR41. This is the same site? No, sorry, I'm sorry, it's confused. So this one is, so we cross GPR41 knockout mother or GPR41 heterozygous mothers, and then offspring again, so GPR41 positive or GPR41 negative. Then we measured the eosinophils and neutrophils and those in the GPR41 negative and GPR41 heterozygous. And then GPR41 negative shows this canceling of eosinophil infiltration by propionate and those. So this means that the mother's GPR41, I'm sorry, I explained maybe differently, but the mother's GPR41 knockout could affect something, but it is not the case that even the mother's GPR41 heterozygous, then offspring of GPR41 negative offspring, yes, so this is a GPR4 the heterozygous offspring for GPR41. So GPR41 knockout is not important, but I mean, in the mother is not important, but the offspring GPR41 knockout is important here. And then we further asked what the propionate mechanisms. So we asked first small intestinal lamina propria cells and lung cells, which cell population subset express GPR41. This is the QPCR, so gene expression. And we found that eosinophils most strongly express GPR41 genes. And then we did the RNA-seq analysis of the control and propionate fed mouse of small intestine. Then we found that toll-like receptors 2, 8, and 9 is highly induced upon propionate treatment of eosinophils, but it is cancelled by GPR knockout, both in lung and small intestinal lamina propria. So this means that eosinophil get the signal of propionate through GPR41, and then these genes are highly expressed. So this is just the summary. So in the neonatal breastfed period, when propionate there sufficient in the gut, then in small intestinal lamina propria as well as in the lung, the eosinophil receive this propionate signal through GPR41 to upregulate TLR genes, so likely TLR signaling as well. And we don't know the exact mechanisms how this suppress the airway inflammation, but not in the neonatal period, but later on in the human case 5 years old, and in mouse 5 to 6 weeks old, the allergic airway inflammation induced by HDM in case of mouse is somewhat suppressed. So we thought that we think that some epigenetic predisposition in the young period could continue in the eosinophil. So eosinophil can be educated by the propionate in the young stage, and then it could affect to suppress the later on allergic airway inflammation. Okay. So these are the collaborators. Sorry, this is Japanese. So not only my group, but laboratory metabolomics group led by Dr. Arita, and laboratory for microbiome sciences led by then Dr. Hattori, and now Dr. Suda, and of course the Department of Pediatrics in the Graduate School of Medicine, Chiba University. Professor Emeritus Shimojo led this group to do this nice cohort. And Professor Ikuo Kimura kindly provided us with the GPR41 and GPR43 knockout mice. And these are funding agencies. And this is a group photo of my group. Thank you very much for your attention.
Thank you, Hiroshi. And I really enjoyed a new story about asthma. Again, thank you very much to joining this global IMMUNOTALKs, and well, the people gonna make a question through the Twitter, so I want you to answer to this those question. But before closing, I would like to explain how to make a question for the audience. Hiroshi, could you stop the
Yeah.
sharing now? Okay, and I'm gonna share one slide. Hope it's working. Yes. So, if you want to make a question to Hiroshi, please go to the Twitter and search for the account Global Immo Talks and find the tweet that said "Ask question for Dr. Hiroshi Ohno here," and reply to the tweet with your question and mention hashtag Global Immune. So that's all for today, and thank you very much for joining us. Hiroshi?
Thank you.
Thank you very much. Okay, have a nice day.